We are now going to begin to talk about biotechnology. We're going to focus on advanced DNA and biotechnological concepts. First, we will discuss polymerase chain reaction, PCR. PCR is a lab technique used to make many copies of a specific piece of DNA. It's like making clones of a tiny DNA section. How PCR works. Denaturation. The DNA sample is heated to separate its two strands. Annealing. The temperature is lowered and primers, short DNA sequences, attached to the specific regions of the DNA you want to copy. Extension. DNA polymerase adds new nucleotides to build the new DNA strand, starting from the primers. These steps are repeated many times, doubling the DNA with each cycle. Why is PCR useful though? It makes millions of copies of a specific DNA segment in just a few hours. So PCR is used in forensics, genetic testing, and research. Gel electrophoresis. This is a lab technique used to separate DNA fragments based on their size. How it works. DNA is placed into wells in a gel. An electrical current is applied. DNA moves through the gel. Smaller fragments move faster and farther, while larger fragments move slower and stay closer to the start. So why is this useful? It's used for DNA fingerprinting, crime scene investigation, paternity test, and genetic research. So what gel electrophoresis does is it sorts DNA fragments by size using electricity. You are now going to watch the video, A Crime Scene Mystery, the science of PCR, enzyme restriction enzymes, and gel electrophoresis. As you watch the video, answer those questions and be prepared to discuss with the class. Genetic engineering is the process of changing an organism's DNA to give it new traits. In general, how this works is scientists cut out a gene from one organism using special enzymes. They insert that gene into another organism's DNA. The new gene gives the organisms a new trait, like making insulin or pest-resistant crops. An example of this is the CRISPR-Cas9. CRISPR-Cas9 is a gene editing tool that allows scientists to cut and modify DNA at specific locations. CRISPR is a part of bacteria's immune system that helps them defend against viruses. Cas9 is an enzyme that acts like molecular scissors, cutting DNA at a precise spot. This technology can be used to correct genetic diseases, modify crops, and even create genetically engineered organisms. So this is important because we can use this in medication, like making insulin. We can use it in agriculture and research. So, genetic engineering means changing DNA to give organisms new trait. The issue with this that is this a good thing or is this a negative thing? And that's where ethics come into play. Biotechnology is very powerful, but people must think carefully about its safety, fairness, and the impacts that it can have on society and nature. So, when we look over this, DNA biotechnology can do amazing things, but it also raises important ethical questions, meaning questions about right and wrong. We always need to weigh certain key ethical questions. Safety. Is it safe for humans and the environment? Privacy. Who should have access to someone's genetic information? Fairness. Could genetic technology be used unfairly, like in jobs or insurance? Genetic modification. Should we change the DNA of humans, animals, or plants? And where do we draw the line? So, looking at some of these considerations, look at medicine. We could use this to personalize medicine based on genetic information. We could use it for gene therapy. In agriculture, we could genetically modify crops so that they produce more or are resistant to pests. Some of your ethical considerations there are: how do we feel about those genetic modifications? Is there an impact? Are we balancing the benefits versus the negative impacts of those things? And that is what ethics are. And those are things that we really need to consider when using this biotechnology. So, in summary, DNA is the material that carries genetic information in living things. Scientists used to think that it was carried in proteins, but experiments by people like Griffith, Hershey, and Chase showed that DNA is the genetic material. DNA is made up of subunits called nucleotides. Those nucleotides are made up of a phosphate group, a 5-carbon sugar, and a nitrogenous base. The 4 bases are adenine, thymine, cytosine, and guanine. Remember, adenine always pairs with thymine, and cytosine always pairs with guanine. The shape of DNA is in a double helix. That is 2 strands twisted together. DNA replication happens before a cell divides, copying the DNA so that each new cell has the same genetic information as the original. During replication, enzymes like DNA helicase, DNA polymerase, and DNA ligase help unwind, add new nucleotides, and then join DNA fragments. Biotechnology like PCR and electrogelphoresis can help scientists study and analyze DNA. Well, genetic engineering uses tools like CRISPR to edit genes. Understanding DNA and these techniques helps us learn more about genetics and the effects on our lives.